Today, Chelsea and myself, Victoria, will be discussing the maternal temperature, corticosterone, and body condition as mediators of maternal effects in Jacky dragons. And this will be based off of the article written by Gracie Liu, Crystal King, and Lisa Schwartz.
The environmental conditions that mothers face have behavioral and physiological effects that can alter offspring characteristics. Factors that influence maternal effects are capable of adapting in various environments and include body condition, diet, thermal regime, stress, predation risk, and nesting phenology. All organisms deal with temperature changes in their environment. These changes can cause physiological constraints on an organism and alter life history traits of the next generation. Ectotherms are heavily influenced by this environmental condition since their metabolic and reproductive process are linked to the temperature of the external environment. External environmental temperatures can directly, through viviparous species, or indirectly, through oviparous species, affect offspring phenotypic traits and temperatures at which offspring are developing. For example, studies showed that when altering the environmental temperatures of reproducing Jacky dragon females, Amphibolurus muricatus, to certain extremes, offspring growth, development, and sex determination was altered. However, ways by which thermal information are sent to offspring are undetermined. Reason for this study was to analyze corticosterone as the potential mediator of thermal maternal effects. This study used the oviparous lizard Jacky dragon to evaluate clutches under different temperature treatments. A captive breeding colony was studied which consisted of a variety of wild-caught and laboratory-born individuals greater or equal to 2 years old. Breeding Jacky dragons were exposed to 2 basking treatments within 2 breeding seasons, 2015 and 2016. Jacky dragons were subjected to either a short bask treatment, which is 7 hours, or a long bask treatment for 11 hours under a 40-watt incandescent basking lamp. With the lamp on, temperatures in each cage ranged from 24 to 57 degrees Celsius. Each group of reproducing individuals were randomly assigned one of the 2 treatments when beginning the 2 breeding seasons. Levels of corticosterone were measured using corticosterone assays, and statistical analysis was conducted on R. Results showed that levels of corticosterone were significantly higher in long basking females compared to short basking females. There was no correlation between body condition and level of corticosterone at the beginning of the breeding season. However, corticosterone levels displayed a negative relationship with body condition in long basking females during the post-breeding season. As females approached their lay date, corticosterone increased linearly in individuals who laid their eggs naturally rather than induced egg laying. Female corticosterone levels in basking treatments had no effect on clutch lay date, hatch time, clutch size, total egg output, or total egg mass. However, female body condition had a positive relationship on hatch time, clutch size, total egg output, and total egg mass. Initially, egg mass was negatively related to female body condition, but differed with basking treatment. As body conditions improved, short basking mothers produced smaller eggs compared to long basking mothers. Also, wild-caught females produced more offspring than laboratory-born females. Maternal corticosterone levels did not explain offspring's sex. However, mass of male and female hatchlings were influenced by corticosterone levels in both treatments. Mothers with high levels of corticosterone produce lighter hatchling males compared to those with lower levels. Overall, there was a stronger positive impact on hatchling mass in longer basking females compared to shorter. Overall, this study signifies the connections between maternal thermal environment, levels of corticosterone in reproducing Jacky dragon mothers, and body condition, and how they influence maternal reproduction and offspring traits, resulting in potential fitness consequences. Thermal regimes alter corticosterone levels, which in turn affects offspring size. Similarly, maternal body condition and environmental temperatures of reproducing mothers strongly influence reproductive output and offspring size. Therefore, this study states that environmental temperature, female corticosterone levels, and body condition are mediators of maternal effects. Although these parameters are highlighted as mediators of maternal effects, conducting further research on offspring traits would be beneficial for assessing their relative roles. Now I'll pass it off to Victoria.
Is this study novel? This study, completed and published in 2020, is in fact a novel study. Although it uses critical information about the species of Jacky dragons from previous literature dating back to the late 90s and 2000s, this specific study itself is bringing light to new information. It was stated that the mechanisms by which thermal information can be passed on to offspring have been greatly underexplored. And out of the 3 major objectives being tested, it was stated that plasma corticosterone has not yet been studied before in Jacky dragons, nor are there any publications for it. So the authors sought to prove firsthand physiological validation and evidence of corticosterone levels in the blood. This study also takes a look at and tries to prove past inconclusive research about the relationships between natural sex steroid levels and offspring phenotypes, of how natural levels of corticosterone in response to ecologically relevant pressures may influence offspring phenotypes, and how plasma corticosterone levels are being related to the thermal environment, thus proving further that this is in fact a novel study on Jacky dragons. The research questions asked within this study were: 1. Is there any evidence to prove physiological validation of corticosterone levels in the blood of the Jacky dragons? 2. Are plasma corticosterone levels related to the thermal environment? And 3. Is there a relationship among maternal plasma corticosterone, reproductive traits, and the offspring phenotypes? In order to answer their research questions, the appropriate methodologies that were used included the random assignment of one of the 2 basking treatments, either the 7 hours or the 11 hour. Basking times were chosen to reflect the natural variation in basking hours, with the lower basking time representing a daily maximum of the thermal regulation ability of the Jacky dragons. And the lizards were then randomly assigned to either ACTH or the control injection, and this did not differ in means of variation in mass and SBL. They also made sure that the corticosterone levels in raw plasma were quantified using the competitive binding enzyme-linked immunosorbent assay, and all of their analyses were conducted in R. The linear mixed effects models were also used to examine the continuous response variables, and the categorical response variables were examined by using the binomial generalized linear mixed models. By using these methodologies, they were able to answer their research questions that were being asked. In order to present and interpret the data that was found throughout the study, the authors used multiple different figures and tables to present their findings. The data found within the tables and figures used statistical analysis such as ANOVA. Linear mixed effects models were used to examine continuous response variables, and categorical response variables were examined using binomial generalized linear mixed models from the studies that were conducted by the authors themselves. Furthermore, in all the models, the maternal cage and ELISA plate were included as random effects, and the likelihood ratio tests of full and reduced models were used to determine statistical significance of explanatory variables in all the models, which tended to avoid any primary biases within the study. The authors were very truthful when interpreting the results that they found during the study, and all of their graphs and tables included all of their results. The results that were included within this study are the physiological validation, the parental temperature influences on condition and stress, the maternal reproductive investment, and the offspring phenotypes of the Jacky dragons. This study was very well conducted, and it was able to display information that was found in an easy way to understand. One improvement that could be made within this study was to undergo more studies on offspring traits to further assess their roles on how they relate back to the maternal factors of the Jacky dragons. It was stated that within this study, the maternal corticosterone levels actually did not explain the offspring sex. So further research on this topic would be beneficial to all scientists and people who want to further their education in the Jacky dragons. It would also be interesting and beneficial to take a look at the paternal roles and how the father's side affects the offspring traits, and how they would provide a further understanding on the offspring itself, including the other half of where the offspring had come from, could provide a lot more information on the Jacky dragons. Chelsea and I want to thank everyone for listening to our presentation on the maternal temperature, corticosterone, and body condition as mediators of maternal effects in Jacky dragons. Thank you.